Hey everybody, Mr. Murphy here. I'm going to run through the entire process of photosynthesis. So the picture that you're looking at here is a pretty good overview. I think it does a really nice job at giving you like the right amount of detail to understand this, as we previously have in an earlier biology class, but also introducing some of the elements of what as AP students we want to begin to focus on and just understand a little bit better. Because that's really what this unit is about. It's about taking something that on its face is not terribly complex, the big goals of photosynthesis, taking light, water, and CO2 and producing sugar and oxygen as a byproduct. And even looking at some of the crazier diagrams in intro level biology class, like we can understand sort of like the big steps of the goals that are happening, but I don't think that we often walk away from that with kind of a real appreciation for the nuance of just how these different things have to happen the way they happen, not just because of like chemical rules that we've talked a lot about, but also because it's fitting into a much larger and very important cycle of energy between different organisms on the planet. Because if this were a reaction which has, a continual buildup of waste, it's not something that's really going to work in a world where organisms are thriving for billions of years, or populations are thriving for billions of years. And so that's what's kind of cool is understanding photosynthesis and respiration together, which requires a little bit of diving into the details. So looking at this, we see it split down the middle, just so we understand how the diagram's sort of lying to us. This is just a chloroplast. This is not showing us a plant cell. Of course, there would be many, many chloroplasts inside of a single leaf cell from a typical plant. And then within that chloroplast, we would actually see tons and tons of these grana, which are these stacks of thylakoids. So they would be all over the place. And the stroma then is just sort of the cytoplasm-y liquid component inside of the chloroplast, and that's where we see the Calvin cycle happening. So when you look at a diagram like this, we see light, we see water coming in, starting on the left side. We see that the thylakoids are responsible for this reaction because these inputs are coming in. Oxygen is coming out from here, and the other thing that's coming out are these energy carriers, ATP, which we're familiar with, and NADPH, which is another electron-carrying, high-energy molecule that is basically enabling other reactions to utilize its energy down the road. So that's the goal of the left-hand side, is to take light and water and to produce these high-energy carriers. In order to do that, it needs the depleted versions of those, right? So I always use the analogy of thinking about ATP and NADPH as kind of like a charged up battery, and then these would be like the dead batteries coming back from the Calvin cycle. This also helps us to understand why it's the light-dependent side, and then the Calvin cycle is often known as the light-independent side. So the energy source for the things that are happening on the left-hand side is the light. It's the photons of light that are striking the chlorophyll within the thylakoids. And the energy on the right-hand side, though, is purely from these energy carriers. So the whole thing is we're charging these energy carriers using energy from light in order to send them off to actually do sort of the main goal of photosynthesis, which is to produce glucose. Now recall from talking in class, what's actually produced is a molecule called G3P, which is glyceraldehyde 3-phosphate, maybe. The point is that it's a 3-carbon molecule, which we'll need 2 of to then convert into a glucose. But thinking about glucose as the goal of photosynthesis is correct, because photosynthesis is the way in which plants and autotrophs in general are producing their own food. It's not making immediately usable energy. Notice that the ATP and the NADPH, they all get used up as directly. That whatever's produced on the thylakoids is used up in the Calvin cycle and then sent back in its depleted form. So this is not how the plant cell is actually getting its energy directly. It's how the plant cell is converting light energy into a chemical storage form, which is glucose. The glucose then is going to go on to be a part of cellular respiration, where it'll actually become that useful cellular energy. Looking at the light-dependent reactions a little more closely, we see this diagram of the thylakoid membrane. That's what this is across the middle here. So this is outside of the thylakoid in the stroma of the chloroplast, and this would be inside of the thylakoid. There's a lot to take in all at once, and you can start at various places. I like to start with Photosystem 1 just because it kind of gives us two directions to understand a little bit better why this whole thing has to happen sort of in concert in order to occur correctly. And photosystems are basically collections of proteins and chlorophyll. And that chlorophyll is going to be the molecule that becomes excited when it is struck by certain wavelengths of light, and it absorbs those wavelengths. And so here in Photosystem 1, we see the absorption of a photon, of many photons, which the energy that's being absorbed there excites electrons in chlorophyll. It's kind of a special molecule in that way that it allows its electrons to become excited and to zip off and do something else. And so when that happens, a couple things occur. First off, that electron becomes very useful. This electron that becomes excited here goes on and bounces between some different proteins and ultimately gets captured by one of our depleted batteries, our NADP, which captures that electron and a nearby hydrogen ion, a proton, and becomes NADPH. And it's now charged up and ready to go. It's already in the stroma. It can now be used to facilitate the further transference of that energy from that excited electron to the NADPH to whatever is going to be happening in the Calvin cycle. The other thing that's happening at the photosystem is that every time it loses an electron, it becomes positively charged, which is problematic for that chlorophyll because if it continues to become positively charged, it's no longer going to be chemically able to just give away electrons. And so what that means is that Photosystem 1 needs an input of electrons for every electron it's losing. And if we look at the dotted blue line here, we see this is a flow of electrons. And so if we sort of run this backwards, we can find out then that the electron that's being replaced in Photosystem 1 is actually coming ultimately from Photosystem 2. So if I back this up now, Photosystem 1 is all about making NADPH using the electrons excited by light. Photosystem 2 does the exact same thing. Light comes in, excites electrons, and the electron from Photosystem 2 is actually bouncing around a few different proteins. These proteins are facilitating basically a proton pump. They are pumping hydrogen from the stroma into the thylakoid, which is beginning to establish a higher concentration of protons, of hydrogen ions, inside of the thylakoid compared to the outside. That kind of fits with what we saw before because NADP is also grabbing a hydrogen and taking it away as NADPH. So this side is sort of doubly losing hydrogens as they are pumped across using the high energy electron from Photosystem 2 and as they are sequestered by NADP and the high energy electron from Photosystem 1. Now, just like with Photosystem 1, Photosystem 2 is going to be losing its electrons, right? It's giving them away as it becomes excited by the photons. And now we can go back to the true origin of the electrons in the electron transport chain of the light-dependent reactions of photosynthesis, and we see that it's actually coming from water. And this is truly why plants need water in order to perform photosynthesis. They need water to be an electron donor. And so as the photosystem 2 is losing electrons, it becomes more positively charged. And there's an enzyme here which is facilitating the splitting of water to enable the electrons to be replaced in photosystem 2. Now, as a byproduct to that, there's two things that are produced. When you break water and you take away its electrons, you're going to be producing oxygen atoms, which can join together to form O2. Okay, that's a nice little side bonus of photosynthesis. It's not useful to the chloroplast, but of course we know in the bigger sense, oxygen becomes a very important factor in how photosynthesis plays into cellular respiration. So we'll see that come back. The other thing we're producing is more hydrogen ions. And remember, we are losing hydrogen ions on the outside of the thylakoid by pumping them across into the thylakoid, and we're also now splitting water, producing even more of these hydrogen ions, these protons, which means we're really building up a high concentration of hydrogen ions. So, if we look at this, photosystem 2 is all about pumping hydrogen ions, photosystem 1 is all about making NADPH. The flow of electrons is from water to photosystem 2 to photosystem 1 to the NADPH. Ultimately, those electrons will end up in the glucose, and then we'll see how their adventures continue in cellular respiration. Spoiler alert, they actually end up back inside of water. And so it finishes its complete cycle. But where we're at right now, we haven't made the ATP yet. So we have this very high concentration of protons right here on the inside of the thylakoid, low concentration on the outside. So there is a concentration gradient established, high to low. And those protons are able to travel through an enzyme called ATP synthase. They can't go straight through the membrane because they're positively charged. They're just protons. So they want nothing to do with the hydrophobic region. They're much more attracted to the hydrophilic environment of the inside of the thylakoid and the outside of the stroma. So they need this enzyme to facilitate their transport across the membrane. And as they do that, this thing literally spins. It's like a revolving door. The charged particles that are traveling through it cause its shape to change, which results in different components inside of this protein shifting and spinning, which then facilitates ADP and a nearby phosphate group from being basically forced together into ATP. So I always joke that ATP synthase it's like a revolving door. It's mooching the energy of whoever's pushing their way through it in order to maybe power some device that's plugged into it. Well, that's essentially what's happening here. It's mooching the energy of these hydrogen ions that are passing through it in order to make ATP. So we have then established our goal. We've reached our goal of making our two energy carriers, NADPH and ATP. We've also produced a waste product of oxygen, and we did that all just by using light energy and the and water coming in, as well as the depleted versions ADP and NADP. Looking at the Calvin cycle, it's a little simpler. Different diagrams of the Calvin cycle can certainly be far more intimidating than this one, but basically what matters is that the Calvin cycle is taking in CO2, is modifying the molecules that are getting these carbons added onto it. Some of that requires energy, so we need ATP, we need NADPH. That's coming from the light-dependent reactions, ATP over here on the left as well. And ultimately, through the chemical reactions, some of which are requiring energy and are phosphorylizing things, you see little phosphate groups being added and removed in various places. Ultimately, we are producing a net gain of one G3P molecule. And remember that two of those will add up to a glucose being produced. The rest of this is really just about how this is able to happen. We can't just take carbon dioxide, throw it together and have, half a glucose. Instead, there's a 5 carbon molecule that scoops up one CO2 and becomes a 6 carbon molecule. That's unstable, so it breaks into 2 3 carbon molecules. Some changes are made to them. Hydrogen bonds and OHs and things are moved around and changed in their shape in order to produce this precursor to glucose, this sort of 3 carbon thing that is ready to be attached to another one to become our standard 6 carbon C6H12O6 carbon glucose molecule. And so for every resulting sort of spin of this wheel, we are producing one extra G3P molecule. The remaining ones end up being recycled, which is really just basically changing their shape using some energy to put them back into this 5 carbon molecule form in order to be ready to get the next CO2. So that's the Calvin cycle. There's an enzyme called Rubisco that's helping to capture that CO2 that has basically dissolved from the air into the tissue of the plant cell and is now captured and turned into this physical form, which is glucose. So that is photosynthesis. It's a lot, I know, but watch this again or go back or check things, and please do ask questions. Thanks.